All right. Hello. Today we will be covering chapter 3, which is over the cell. Um, as always, we'll start out with some roots, prefixes, and suffixes that you'll need to know for this chapter. Let's see. Just something to point out, endo means within, hyper means above, hypo means below, iso, like an isosceles triangle, means equal, lyse means to break up, phag, like phagocytosis means to eat, pino means to drink. So just keep some of these in mind as we move through the chapter. All right, so cells. As we learned in chapter 1, the basic unit of structure and function in the body. We measure cells in micrometers. Don't really need to know that. Some cells move independently, and some cells are fixed. They don't move. For example, your red blood cells, your white blood cells, your platelets, they're going to be moving around in your cardiovascular system throughout your bloodstream. Your liver cells, they stay in your liver. Your kidney cells, they stay in your kidney. Your small intestine cells, they stay in your small intestine. They don't move around. Cells have developed specialized characteristics. They're said to be differentiated. We talked a little bit about this in a previous chapter that, all cells have the exact same DNA, but due to genetic expression or differentiation, certain cells act as a kidney cell. A kidney cell cannot be a liver cell. A small intestine cell cannot be a large intestine cell. A neuron, a nerve cell, can't act as anything but what it is, and it's due to genetic expression. And the goal of everything is to maintain internal homeostasis. We'll see that cells vary in size and shape. Small red blood cells are a lot smaller than smooth muscle cells or skeletal muscle cells. Skeletal muscle cells can be up to like 8 inches in length. And it just allows for different types of cells to perform their unique functions. And it allows for that structure, that structure and function are interrelated. We have about 50 to 100 trillion cells in the adult body. All right, so here you can see just some pictures of cells. This one up here is a nerve cell. If you remember, these are your dendrites, cell body, axon. This is just a little sheet of simple squamous epithelium. This would be something that like lines your alveoli, which performs gas exchange in your lungs, oxygen, carbon dioxide. This right here looks like a cardiac muscle cell. This would be a red blood cell, a white blood cell. This would be like a regular cell. This is a smooth muscle cell. So you can see how all the cells kind of vary in shape and size. They look very different. And like I said, it just allows for them to perform their specialized function. So as we saw, there is no typical cell. Cells really don't look alike. But what we find is that cells, for the most part, have 3 common parts. They all have a cell membrane or a plasma membrane. They all have a nucleus and they all have a cytoplasm. Now, the cell membrane, I think, as you should know, is that outer boundary. It maintains the cell's integrity, and it controls the entry and exit of substances. It will allow sodium, potassium to come in. It will allow calcium to come in, a lot of ions. a lot of chemicals. The nucleus. Remember, inside the cell is that nucleus. And the nucleus contains DNA. It contains that genetic material, and it's going to direct the activity of that cell. Now, there is an exception. Red blood cells do not have a nucleus. So just keep that in mind. Cytoplasm. Cytoplasm is kind of like the all of the inside of the cell. We have the gel-like fluid called the cytosol, and then we have a bunch of organelles that we'll talk about in this chapter. You know, the nucleus, the mitochondria, the endoplasmic reticulum, the ribosomes, the scaffolding like the microtubules, your Golgi apparatus. All of those are organelles, and they're suspended in that gel-like fluid called cytosol. And each organelle has a specific function, and some organelles dismantle cellular debris or bacterial debris or viral debris. Some organelles extract energy. I think we all know that the mitochondria is the powerhouse of the cell. So, you know, that's the organelle that's responsible for extracting energy. Or some organelles package secretions or package proteins. So the organelles are actually the labor units of the cell. All right, so here's a typical composition of a cell. But just know that that this isn't always how the cell looks. If we take a picture now versus 5 seconds from now, these organelles, they move around. The cellular membrane isn't rigid. It's very flexible. It allows for movement in between tight spaces. So the cell will kind of swell up and shrink down because it has to allow for stuff to come in and out. So you can just see some of the organelles in here. We have the mitochondria, this Golgi apparatus. We have some smooth endoplasmic reticulum. We have some rough endoplasmic reticulum. We have the nucleus here. We have a nucleolus. We have some ribosomes here. We have peroxisomes. There's your cell membrane. Remember that cell membrane is made of that phospholipid bilayer. We talked a little bit about that last chapter with phospholipids. Let's see. Some cells, like your sperm cells, have this flagellum. It allows sperm cells to move around. And then we'll talk about the centrioles. So there's just a lot of organelles within the cell. And some cells have more organelles depending on their function. So a skeletal muscle cell might have more ATP than another cell, or an athlete who runs track or who's a sprinter might have more mitochondria than someone who's a couch potato, because they're going to need more energy or more mitochondria to produce that ATP or that energy than someone who is a couch potato. So some cells will have more organelles than another cell or than another person, depending on the activity level or what that cell needs. All right, so the cell membrane. Obviously, it's that outer boundary. It maintains the integrity of the cell. It's very thin, very thin. Look, you know that phospholipid bilayer. It's very thin. It separates that intracellular fluid, what's inside the cell, from the outside, that extracellular fluid, or that fluid that's outside of the cell. And remember, that fluid that's inside of the cell is cytosol. It's very gel-like substance. So cytosol is the gel-like substance. The cytoplasm is the cytosol plus all of the organelles. And we say that the cell membrane is selectively permeable in that it regulates the entry and exit of certain substances. Not everything can come in and out of the cell. And it just depends on the size of the molecule, the polarity, and other factors. So obviously, if your cell is this big and you have a molecule that is this big, obviously that molecule can't get into the cell. And we talked a little bit about polarity last chapter. Polarity is polarity will also help determine whether or not certain substances can or cannot enter into the cell. Signal transduction permits cells to receive and respond to messages. We have cell-to-cell communication. Cells have to be able to communicate with one another. We'll talk about this with smooth muscle cells in the digestive system. We have this thing called peristalsis that moves food through the small intestine, through your alimentary canal. Cells have to say, okay, I contracted, now it's your time to contract, now it's your time to contract, now it's your time to contract. And that's how food gets moved through the small intestine, or also through your esophagus, through your large intestine. Same thing with the heart. We have these things called intercalated discs that you want the heart to contract in unison. So you want the atria to contract, and the ventricles, and the atria. You don't want your heart cells to just randomly contract without some sort of order. That's actually called ventricular fibrillation, when cells just randomly fire and contract. And then if you watch, any type of medical drama, oh, they're in VFib, we need to shock them. So you want your heart cells, your cardiac cells, to be able to talk to one another. That cell membrane consists of a lot of lipids and proteins, and it does have some carbohydrates in with it. And we'll see a picture of the cell membrane with all of this stuff here in a few slides. All right, so just generically, we have the cell membrane that consists of phospholipids. We call it this phospholipid bilayer because we have kind of two phospholipids, one on each side, that separates the intracellular fluid from the extracellular fluid, the inside of the cell versus the outside of the cell. And again, it's responsible for the membrane flexibility. The cell has to be able to squeeze through tight surfaces. We have a water-soluble side and a water-insoluble side. Hydro means water, Phil. Philic is the adjective form. Hydrophilic means to love. Hydrophobic means to fear or to hate. You have a phobia. So we have water-soluble or hydrophobic heads. So these heads are going to be oriented towards the sides with water. So your intracellular and your extracellular sides of the bilayer are going to have your water-philic heads. So like your examples of water-soluble compounds or water-soluble molecules are going to be amino acids, sugars, protein, nucleic acid, and various ions. So, which makes sense because if you take sugar and you dump it in water, sugar is going to dissolve. If you take sodium and chloride, sodium's an ion, chloride's an ion, that's table salt. You take table salt and throw it in water, you're going to throw it in water and it's going to dissolve. Sugars. Basically, sugar is a carbohydrate. Carbohydrates get processed down to sugar. So you take a piece of bread and you throw some bread in water and stir it up, and eventually it's going to dissolve. Water insoluble just means that it can't be dissolved in water. So these are going to be, if you remember the structure of a phospholipid, we have that glycerol backbone, 2 fatty acid side chains, and a phosphate group. So these 2 structures right here are going to be your fatty acid side chains. Remember, water and oil, they don't mix. So water and fat, they're not going to mix, which is why those fatty acid side chains are pointed towards the inside of that phospholipid bilayer. So some things that are water insoluble or hydrophobic: oxygen, carbon dioxide, steroid hormones. Remember, cholesterol is a lipid. So anything that is a lipid is not going to be soluble in water. So this bilayer is actually permeable to lipid-soluble substances, but not to water-soluble substances, meaning that a lot of lipids can cross this membrane, but water-soluble substances cannot, because this membrane is mostly made up of lipid structures, these fatty acid side chains. All right, if we look at the structure of the cell membrane, we'll see that a lot of, we know that phospholipid bilayer is present, but we see that there's proteins molecules that are embedded in this cell membrane. And we say that it's a fluid mosaic, has fluid mosaic properties. And that just means that just as the cell can change shape, to fit tight spaces, that whatever's in this cell membrane also can move around. Just because a protein is, right here in a cell membrane doesn't mean it's always going to be right here in a cell membrane. These proteins are going to move around. So just some examples of proteins. We have these pores, channels, receptors, enzymes, cellular contact receptors for identification, cellular adhesion molecules. We'll talk about these different types of receptors later on. They're going to have a lot of different functions. They have different shapes. Some are globular, some are rod-like, some are fibrous structures. We'll see cholesterol in the cell membrane that's going to help stabilize the cell membrane. And remember, cholesterol is a fat, so it's going to help keep that cell membrane impermeable to water-soluble substances. Remember that cell membrane is impermeable to water-soluble substances, but permeable to lipid-soluble substances. Cell membrane also has some carbohydrates involved in the cell membrane. And its carbohydrates are basically used for cell recognition, self-markers. All right, so here you can see kind of a visual representation of those cell membranes. And remember that this is a fluid mosaic model, so that all of whatever you see, they're going to move around within the cell membrane. So first, these channel proteins right here. We're gonna call these a lot of different things throughout this course. And these are going to allow ions to go through. We can call them, channel proteins. We can call them ligand proteins. We can call them ion-gated proteins. Eventually, we can call them voltage-gated channels. We'll call them a lot of different things. We have these receptor proteins. Those are just going to be, as the name suggests, receptor proteins with carbohydrate chains on them that allow the cell to have some sort of receptor on them. They could be for cell recognition. Hey, I'm a red blood cell. Hey, I'm a white blood cell. Hey, what am I? We'll have some cholesterol that is embedded within the cell membrane, again, to help with lipid solubility or water insolubility. We'll have these integral proteins. We'll have peripheral proteins. And we'll talk about the cytoskeleton when we talk about organelles. Again, cellular identity markers, those are also a type of receptor protein. There's another integral protein. Now, one thing to remember, or one thing to know, is that these peripheral proteins, they don't cross into the cell membrane. Integral proteins, they cross into the cell membrane. Transmembrane proteins actually cross all the way through the cell membrane. So you're going to have like a quiz question or a test question that says something like, I can't remember the exact wording, but it's like integral proteins just partially cross into or span the bilayer. Transmembrane proteins go all the way through the membrane. So basically, all transmembrane proteins are a type of integral protein, but integral proteins are not transmembrane proteins, and that they don't go all the way through the membrane. So a protein that spans the cell membrane is going to be classified as an integral protein. And in this case, span would mean to cross into just a portion of. All right, so here's just a summary slide. Types of membrane proteins. So we have receptor proteins. They're going to respond to extracellular signals. These pores, carriers, channels, they're going to transport small molecules, ions, transduce signals, enzymes. Remember, enzymes catalyze chemical reactions. Cell surface proteins, they're going to establish the cell. And I said that later on we'll talk about cellular adhesion molecules. As the name implies, they enable the cells to stick to one another. All right, so cellular adhesion molecules. We also call them CAMs. They are a type of peripheral protein, and they're going to help cells that are kind of within the cardiovascular system. Help cells that are on the move. So we have selectins. Let me find my little pointer. So several types of cellular adhesion molecules, selectins, adhesion receptor proteins, integrins. Then we have another kind of classification called other cellular adhesion molecules. But so let's say that you puncture your, you get a little splinter in your finger. So what would happen is, let's say you get a splinter in your finger. So what would happen is your white blood cells, we actually call those leukocytes. They're already present in your bloodstream, and they're going to be the ones to fight off the infection. We'll talk about that next semester. So once a splinter gets into your hand, a bunch of things are going to start to happen. And we're not going to go into what really happens now, but we're going to see, clotting blood, bacteria, and decaying tissue at this injury site. And that's going to release a lot of biochemicals, these things called chemoattractants that attract the white blood cells to this infection site in your finger. And when the white blood cells get near this splinter, they need to start slowing down in this fast-moving cardiovascular bloodstream. And a type of cellular adhesion molecule, a type of CAM called selectin, slows down the white blood cell by coating the white blood cell. And this is going to provide traction and friction. The white blood cell then kind of slows to a roll, and it's going to bind to this adhesion receptor protein on the inner capillary surface. And then another type of CAM called integrin contacts an adhesion receptor protein that protrudes into the capillary surface near that splinter. And the integrin then pushes up through the capillary cell membrane, and it kind of grabs that white blood cell and it directs it between these tile-like cells of the capillary wall. And white blood cells that accumulate at the injury site then produce a bunch of inflammation, and then that dying bacteria form pus. And then when we get to one of the chapters next semester, we'll kind of figure out what happens from there. All right, so we talked a little bit about some faulty channel diseases. Last chapter with these abnormal chloride channels with cystic fibrosis that traps salt inside, and because of that traps that abnormal chloride leads to pulling in sodium, and wherever sodium goes, water follows inside the lung passageways, and we have to do that chest percussion to get rid of all of that gunk. But we also have mutations in sodium channels, which actually could result in the inability to feel pain or cause patients to feel extreme pain conditions. And we'll revisit this in Chapter 9. Mutations in calcium channels, whether we have hyperkalemia, high potassium, or hypokalemia, low potassium channels, or low potassium levels, that could actually disrupt the electrical activity of the heart and cause arrhythmias. Arrhythmia is just another term for abnormal heart rhythm. So for you future nurses, abnormal potassium is something to be very concerned about. All right, cytoplasm. We've really already gone through cytoplasm. Cytoplasm is what's inside the cell. Remember, cytoplasm equals your cytosol plus your organelles. Your cytosol is the gel-like fluid portion of the cytoplasm, and the organelles is all the working units of the cell. Inside that cytoplasm, we have that cytoskeleton, which is a bunch of protein rods and tubules. We'll briefly mention those later on. All right, ribosomes. It's the first organelle that we're going to talk about. It's composed of protein and RNA. ribonucleic acid, not DNA. Remember, DNA is in the nucleus. So protein and RNA. It's suspended in the cytoplasm, or it is on the rough endoplasmic reticulum. Ribosomes do not contain a cellular membrane, but what they do is they provide a lot of structural support and enzyme activity to build up proteins. They link amino acids to make proteins. Remember, amino acids are the building blocks of proteins. So ribosomes kind of help build proteins. So anytime you think of ribosomes, think of amino acids and protein synthesis. So if we go back kind of all the way up here, we see our rough endoplasmic reticulum right here. And you can see the purple dots that are on this rough endoplasmic reticulum. So they're on the rough endoplasmic reticulum, or they are free floating in the cytoplasm. All right, endoplasmic reticulum. Basically, the endoplasmic reticulum, they're just a bunch of membrane-bound flattened sacs. They look like cylinders or canals or bubble-like sac vesicles. They're a network of interconnected membranes. Basically, they start at the nuclear envelope and they go all the way to that cell membrane. And they connect with a bunch of other organelles. Basically, they're a transport system. We have two different types of endoplasmic reticulum. We have a rough endoplasmic reticulum and a smooth endoplasmic reticulum. The rough endoplasmic reticulum we saw contains those purple dots. They contain ribosomes. And that rough endoplasmic reticulum, remember ribosomes, protein synthesis. So the rough endoplasmic reticulum is going to be responsible for protein synthesis. And those ribosomes that are attached to those rough endoplasmic reticulum will eventually go to the Golgi apparatus, which we'll talk about next in a couple slides. We'll go to the Golgi apparatus later on. So that rough endoplasmic reticulum responsible for packaging protein molecules for eventually secretion. And we'll talk about I'll talk about it now. So secretion doesn't necessarily mean secretion outside of the cell. It could mean that, once we package our proteins, it could go outside of the cell for somewhere else in the body, but we also have ribosomes that were free floating in that cytoplasm. It could be to go secrete back into the cell for some sort of intracellular work. All right, so smooth endoplasmic reticulum. We don't see any ribosomes on the smooth endoplasmic reticulum, but what it does is conduct lipid synthesis. We call that lipogenesis. Basically, we're producing fat. Lipo is fat, genesis means to create. So lipogenesis is basically converting excess dietary carbohydrates to fats. So you eat too much carbs, you're going to get some belly fat. That's lipogenesis. All right, so here is the structure of the smooth and rough endoplasmic reticulum. Here you can see the rough endoplasmic reticulum. You can see the ribosomes on there. Here is the smooth endoplasmic reticulum. Here's the rough endoplasmic reticulum. You can see all the purple dots on there. They both have another phospholipid bilayer. Remember, ribosomes, they do not have a membrane. All right, so Golgi apparatus. Remember that ribosomes from the rough endoplasmic reticulum for protein synthesis go from the rough endoplasmic reticulum to the Golgi apparatus. The Golgi apparatus, basically, they're again flattened sacs. We call those cisternae. Basically, they're going to refine, package, and deliver the proteins that were made on the rough endoplasmic reticulum. And remember that that I said that that the rough endoplasmic reticulum, or that those proteins are going to be eventually be secreted. Not necessarily secreted outside the cell, but it could be to go into, to be used within the same cell. And then they are packaged within these vesicles. Vesicles, basically, it's not really showing these correctly. Basically, they're in a sphere. It's not like that they're in this cup that this picture is showing. They're in a sphere. So they're kind of enclosed in a ball. So vesicles, we'll see vesicles later on in the nervous system chapter and the muscular system chapter. Basically, vesicles are just membranous sacs, and they're going to either store or transport substances within or between cells. And if we are secreting something outside the cell, basically these vesicles will fuse to the cell membrane, and then the contents of the vesicle will then be dumped outside of the cell. Some of these vesicles will contain water. Some of these vesicles, in this case, as this picture shows, they contain proteins, because that's what's happening from the Golgi apparatus. Some vesicles would contain broken down bacteria. Let's say that a white blood cell is trying to fight off infection and gobbles up a bacterial cell. And now we have this vesicle of a gobbled up bacteria cell, and the cell breaks it down. Now we have a vesicle of a broken up bacterial cell. It might dismantle it, and it might ship it off to somewhere else. It would still fuse to the cell membrane and dump out the gunk of the dismantled bacteria to go wherever it needs to go. All right, on this slide, milk secretion. Really, you're gonna, this is, so for breast milk, basically just kind of memorize the sequence of events that happen when secreting milk. First, things pass through the nucleus, then the endoplasmic reticulum, or the rough endoplasmic reticulum. Then we know it goes to the Golgi apparatus to get packaged into vesicles. Then it's going to, get passed into the cell membrane, fused to the cell membrane. And actually, I lied. Actually, for milk, remember, milk is actually a fat, so it would actually go through the smooth endoplasmic reticulum, not the rough endoplasmic reticulum. So milk fats are synthesized in the smooth endoplasmic reticulum. Most proteins are synthesized on ribosomes associated with membranes on the rough endoplasmic reticulum using amino acids in the cytosol. Sugars, carbohydrates are synthesized in the smooth endoplasmic reticulum and Golgi apparatus, and then they're attached to proteins and secreted in vesicles. So remember, milk is a fat, so fat would go on the smooth endoplasmic reticulum. So nucleus, smooth ER, Golgi apparatus, vesicles, cell membrane. All right, mitochondria. We all know powerhouse of the cell. So each cell has about 1,700 mitochondria. And again, like I said, if you're running track, you're going to have more in your skeletal muscles. You're going to need more energy in the cells that you're going to use more. Basically, these mitochondria have, it's a fluid-filled sac, but they have two membranes. They're going to have an inner membrane and an outer membrane. And you can see that the inner membrane is kind of folded up. This inner membrane is folded, and those folds are called cristae. And basically, the purpose of this cristae is to increase the surface area for chemical reactions. And in next chapter, we're going to talk about those chemical reactions. We talk about citric acid cycle, electron transport chain. That's what happens in the mitochondria. So that produces ATP, it produces energy. This is your cellular respiration. Remember, we digest food and we use that oxygen to extract the energy from the nutrients that we eat. And then obviously ATP or energy is used to power or to provide energy for many of our cellular processes. All right. Lysosomes, peroxisomes, lysosomes, the garbage disposals of the cell. Lysosomes, they're tiny vesicles. They're tiny membranous sacs. And lysosomes, you're going to have to know the difference between lysosomes and peroxisomes. They're almost the same thing, but they do different things. Lysosomes, they're going to digest proteins, carbohydrates, nucleic acids, bacteria, cellular debris, worn out cell parts. And they work by this thing called autophagy or autophagy, or however you want to pronounce it. They kind of eat their own self. Peroxisomes. Lysosomes. So in that example that I said, that we pull the bacteria into the cell and we break down that bacteria. Basically, once we pull the bacteria into the cell, lysosomes would come up attached to that bacteria. It would dump in its contents, and the contents of the lysosomes would actually destroy that bacteria and break up that bacteria. Peroxisomes. They're very similar to lysosomes. They have a membrane, but they are going to digest lipids, fats, alcohol, and hydrogen peroxide. So remember the difference of what lysosomes and peroxisomes do. Lysosomes, proteins, carbs, nucleic acids, bacteria, debris, worn out cell parts. Peroxisomes, lipids, alcohol, and hydrogen peroxide. All right, other cytoplasmic structures are not really organelles, but they're going to help the cell to move or to divide or to transport substances. We have these things called microfilaments. Microfilaments are basically tiny rods of this molecule called actin. They provide cellular movement. So you can see these microfilaments down here. And when we talk about skeletal muscle contraction in chapter 9, we'll learn a lot about actin. Actin and myosin are the two components of skeletal muscle contraction. But when we talk about skeletal muscle contraction, we say that they're part of these things called myofibrils. And the myo portion means muscle. So you're going to have one question about myofibrils. And myofibrils, since myo means muscle, myofibrils are only found in muscle tissue. So they're not going to be found in all cell types. So just remember that. So microfilaments, they contain actin. Microtubules, they're going to be larger than microfilaments. They contain this thing called tubulin. They're more scaffolding-like. They help maintain the cell shape. They make up these things called flagella, cilia, and these things called centrioles. They're going to help move organelles around within the cell. So we'll look at the cilia, flagella, centrioles later on. And then these intermediate filaments, basically they're composed of proteins. They're going to just provide, again, support, more scaffolding, support structures. They're going to support that nuclear envelope, which is your nuclear membrane. All right, so I said that we talk about these centrioles. Centrioles. We have these things called centrosomes. Centrosomes. We have basically, we also call them a central body. They consist of 2 centrioles. They're composed of microtubules, which we talked about on the previous slide. They're in the cytoplasm near the nucleus. They don't have a membrane. So they're organized as 9 groups of 3. So you can see here, there's a group of 3, there's a group of 3, and there's 9 of them. And if we look at them, there's going to be 1, 2 of them. And when we talk about mitosis, that's what these centrosomes are going to be used for. They're going to move to opposite body. They're going to move to opposite sides of the cell. They're going to produce these spindle fiber things, and they're going to pull chromosomes apart for mitosis. But we'll get to that later on. And they also produce these things called cilia and flagella. So cilia is kind of really tiny, these motile or modal extensions of the cell membrane. We have these things called hair cells in the ear, but you can kind of see they're kind of like hair cell. They're kind of like little tiny projections or hairs on top of the cell. More like a fringe on certain epithelial cells. We'll see these in the digestive system. We'll see these in the respiratory system. We'll see these they propel the egg towards the uterus. And what this does is, again, it's in the respiratory system, in the digestive system, they're going to be used for increasing surface area. Or in the digestive system, it's used to increase the surface area so that those food particles can get down into these cilia to increase absorption, increase the surface area for absorption. In the respiratory tract, it's going to help get all of that bad gunk that you breathe in. You know, every time you take a breath, you're breathing in dust and bacteria and viruses and just some bad things. So it's going to help propel that mucus in the respiratory tract back up so you can either cough it out or sneeze it out. Flagella. Really, we'll see this in sperm cells. That's what make sperm cells swim or move. So the sperm cells really only have one flagellum. Flagellum is singular, flagella is plural. We also have microvilli. Microvilli is also composed of actin. We also call that the brush border. This is what we see in the digestive system. We also have these things called inclusions, which we're not going to talk about. So we'll just skip that. All right, some diseases at the organelle level. MELAS, mitochondrial encephalomyelopathy, lactic acidosis, stroke-like episodes. Basically, this is a DNA error in the mitochondria. It's a mutation of your DNA. Basically, you're going to have encephalopathy, headaches, neurological deficits, Crab disease or Krabbe disease. Basically, you have the basically it's a lysosomal storage disorder where you have the inability to produce one of the lysosomal enzymes called galactocerebrosidase. So what happens is, remember what lysosomes do. Lysosomes versus peroxisomes. Basically, these lipids build up in various tissues and destroy brain cells. And when we talk about myelin in one of the subsequent chapters, the person cannot produce myelin. Myelin is going to be wrapped around nerve cells. So basically, it's going to lead to nervous system issues. So I wanted to make you aware, even though that, remember, and I told you, remember the difference between lysosomes and peroxisomes. It's a lysosomal storage disorder. So remember, peroxisomes do lipids and lysosomes do, proteins and other things. So since it's a lysosome storage disorder, that means we're going to have a buildup of lipids. All right, adrenoleukodystrophy, ADL. It's a genetic abnormality. It also affects the nervous system. Basically, we're dealing with peroxisomes or the membrane of peroxisomes. Basically, we can't break down long chains of fatty acids, and eventually those fatty acids are going to accumulate over time. And again, it's going to destroy the myelin sheath. When we look at the nerve, basically on the axon, remember we have that dendrite cell body, and the axon that comes off of the cell body, that myelin sheath wraps around the axon. But this adrenoleukodystrophy is going to attack that myelin sheath, and it's going to eventually lead to brain deterioration. All right, the nucleus. Nucleus contains your DNA, contains that genetic material, and it's going to direct the activities of every cell. Remember, the red blood cell does not have a nucleus. The nucleus has a membrane. We call that a nuclear envelope. It's a double-layered membrane that surrounds the nucleus. Now, inside the nucleus, we call it a nucleoplasm. That's just the gunk that's inside the nucleus, so it separates the nucleoplasm from the cytoplasm. But you can see that we have all these pores in that nuclear envelope, and that just allows for things to go in and out of the nucleus: proteins, dissolved substances, and mRNA. We'll talk about this when we talk about DNA replication. The nucleolus. Basically, it's the nucleus within inside the nucleus. This is the nucleolus. Basically, the nucleolus is a congregation of RNA and protein. This is where ribosomes get produced. There's chromatin inside the nucleus. This is where our chromosomes are going to reside. Basically, and it also stores information for protein synthesis. Um, the when before we do mitosis, our DNA or our chromosomes, they're not, our DNA is just not sitting there in our typical X, Y that we think of. Our DNA is all uncoiled, and we really can't see it under a microscope. And right before mitosis happens, that's when our DNA gets coiled up to where we can see it under the microscope. All right, this is just a summary slide of what the organelle parts, the structure, and the function of the different organelles are. All right, so Anderson's disease results in the failure to secrete large proteins called chylomicrons. Which organelle is most likely affected? So we can't secrete large proteins. All right, so what secretes large proteins? Lysosomes. Lysosomes are responsible for breaking down proteins, not secreting them. The nucleus creates, stores DNA and creates ribosomes. Mitochondria produces ATP. So vesicles are packaged proteins, and they create they have those packaged proteins in the vesicles, and they secrete proteins outside the cell. So that would be the answer that we go with. All right, a person who ran 6 miles a day throughout high school is injured during their first season during running cross country. They must rest for 3 months and then gradually begin exercising again. The skeletal muscles in their lower limbs decrease in size. We call that atrophy. During this prolonged period of inactivity, what organelles that break down muscle proteins are responsible for the atrophy? So what muscles break down, or what organelles break down muscle protein? All right, centrosomes. Centrosomes, remember, are those two things that are going to be responsible for mitosis. They're going to create spindle fibers and pull the chromosomes apart. Peroxisomes break down fats, hydrogen peroxide, lipids. Ribosomes create proteins. Lysosomes break down lysosomes break down proteins, cellular debris, and that stuff. So breakdown proteins are going to be lysosomes. All right, movements inside and out of the cell. So we have passive mechanisms and active mechanisms. Passive mechanisms do not require cellular energy. Cellular energy is ATP. So active mechanisms, which we'll talk about later, require ATP. So in this case, don't require ATP. Diffusion, osmosis, facilitated diffusion, filtration. Diffusion or simple diffusion. Definition: movement of atoms, molecules, or ions from a region of higher concentration to a region of lower concentration. So, this is when you are in the back of the classroom and you spray a perfume. Spray the bottle of perfume in the back of the classroom. The time you spray the perfume in the back of the classroom, you have a higher concentration of perfume in the back of the classroom than you do at the front of the classroom. And eventually, over time, so now you have a concentration gradient. You have a higher concentration of perfume in the back of the classroom than you do in the front of the classroom. And over time, those molecules, those perfume molecules, are going to move all around the classroom, and eventually the front of the classroom will be able to smell the perfume. So at that time, when the perfume has gone throughout the classroom, we say that we've reached equilibrium. So it's the movement of atoms, molecules, ions from a region of higher concentration to a region of lower concentration. So we say that we have that concentration gradient. When we go from a region of higher concentration to a region of lower concentration, we're saying we're going down the concentration gradient. Down the concentration gradient. All right, so those atoms, molecules are in constant motion inside the cell. It occurs only with substances that the cell membrane is permeable to. Remember that the cell membrane is permeable to lipid-soluble substances. So lipid-soluble substances. And if we go back up to, this slide way up here. This bilayer is permeable to lipid-soluble substances, but not water-soluble substances. Oxygen, carbon dioxide, steroids. Steroids are fats, urea, ethanol, anesthetics, help put you to sleep. Outside of the cell, a sugar cube dissolving in water. It's an example of simple diffusion. You put a sugar cube in water, it's going to dissolve. The perfume, simple diffusion. We talked about solute, solvent, and solution. Solute is what is being dissolved. Solvent is what is doing the dissolving. Solution is your final thing. So your solute is a sugar cube. Water is a solvent. Solution is sugar water. And eventually, we're going to reach equilibrium. Once we reach equilibrium, the concentration of water and solute is going to be equal. If in the perfume example, if I take a measurement of the perfume in the back of the class versus the concentration of perfume in the front of the class, they should be equal at equilibrium. All right, so simple diffusion. This is your sugar cube in water. This is your table salt in water. All right, diffusion through a cell membrane. Sometimes, let's say that we have some sort of lipid-soluble substance. Remember, lipid-soluble substances cross the cell membrane. Eventually, over time, we're going to reach equilibrium. If we count the number of red dots on each side of the cell membrane, they should be equal. We see this at the cellular level when we exchange carbon dioxide and oxygen. We breathe in oxygen. Oxygen goes from the lungs to the bloodstream, and in the bloodstream, at the cellular level, we have a higher concentration of oxygen in the bloodstream than we do in the cells. So oxygen is going to diffuse from the bloodstream into the cells, and we also have a higher concentration of carbon dioxide in the cells versus the bloodstream. So carbon dioxide is going to diffuse from the cells into the bloodstream. Then the bloodstream goes back to the lungs, and the reverse is going to happen. We have a higher concentration of carbon dioxide in the bloodstream versus the lungs. So carbon dioxide is going to diffuse from the bloodstream to the lungs. Oxygen is going to diffuse from the lungs to the bloodstream. All right, facilitated diffusion. Facilitated diffusion is essentially the same thing as diffusion, except that we have to have these carrier proteins or these ion channels. Remember I said that ion channels, we're going to call them 47 different things by the time the end of the semester is done. We have these membrane proteins, transporters, ion channels, gated ion channels. Basically, facilitated means it's being helped. We're going to have these carrier molecules on the surface of the cell membrane. It's still a passive process. No ATP is required. We're still going down the concentration gradient from a higher concentration to a lower concentration gradient. And these are going to be used for water-soluble substances that can't diffuse across the cell membrane. Remember, water-soluble substances can't cross the cell membrane because they are not lipid-soluble. So your ions, glucose, amino acids, and this is just my visual representation of the cell. So we have a higher concentration of sodium outside the cell versus inside. We have a higher concentration of calcium and chloride outside of the cell versus inside. We actually have a higher concentration of potassium inside the cell versus outside of the cell. So sodium, calcium, chloride would all diffuse inside the cell. Potassium would diffuse outside the cell. And we would need these carrier proteins or these ion channels or these ion gated channels or these membrane proteins or whatever we want to call them in order to get these ions in and out of the cell. All right, so what affects the diffusion rate? Well, obviously the membrane surface area. The larger the cell, the bigger the cell membrane, the more we can get in that cell. The size of the molecule. Again, if we have a cell this big and a molecule that's this big, we're not going to be able to fit that molecule into that size of a cell. The distance. If I have, using the perfume example, if I have a classroom that's the size of a football field, it's not going to diffuse as fast as if I have a classroom that's the size of a closet. Temperature. Typically, you're going to have a better diffusion at higher temperatures. The concentration gradient or the concentration of the substance. If I only do 1 squirt of perfume, it's not going to diffuse as fast as if I squirt 15 squirts of perfume. So generally, diffusion is going to be more rapid over shorter distances, the closet classroom, steeper concentration gradients, 5 squirts of perfume, and at higher temperatures. Now, why wouldn't, just a question for you, why wouldn't the amount of energy available not fall under here? If I have more energy, why wouldn't this be a factor that influences the rate of diffusion? Because we're talking about passive processes. Passive processes do not require ATP. So the amount of energy available is not a factor in diffusion rates. All right, osmosis. Osmosis, we often call it the diffusion of water. Remember, water obviously is water soluble, not lipid soluble, so it can't cross the cell membrane. So water moves through these things called aquaporins. Basically, they're another type of channel. They're a water channel between that phospholipid bilayer. It's still a passive process. No ATP is required. We're still going to go down the concentration gradient, higher concentration to a lower concentration of water. All right, so now I'm going to confuse you a little bit. Well, not yet. All right, so when we look at the composition of the blood, we're going to see that, and we'll dive into this a lot next semester. We see that about 45% of our blood is made up of these things called formed elements: red blood cells, white blood cells, and platelets. We're going to spend some time on this 55%, our blood plasma. You can see that about 92% of our plasma is made up of water. And then we have, 7% proteins, albumins, globulin, fibrinogen, waste products, electrolytes, nutrients, vitamins, hormones, and our gases: oxygen, carbon dioxide, nitrogen. So basically we have 92% water, 7% protein, and 1% everything else. So 92% water, 7% protein, 1% everything else. So here's where it gets a little confusing, but this is going to be an introduction to next semester. So we have this thing called osmotic pressure. Osmotic pressure is anything that's dissolved in the plasma contributes to osmotic pressure. So dissolved in the blood. Proteins are dissolved in the blood. Proteins contribute to osmotic pressure. Electrolytes are dissolved in the blood. Waste products, nutrients, vitamins, hormones, gases, all of those are dissolved in the blood. So they produce, or they contribute to, osmotic pressure. All of those things contribute to these things called osmotic pressure. We also have this thing called hydrostatic pressure, which I'll talk about here. I'll just jump down to a slide. Hydrostatic pressure, that's the pressure of water. So we have water pressure, which is hydrostatic pressure, and we have osmotic pressure, which is the pressure exerted from anything that's dissolved in the blood. So we have kind of two different pressures in the blood: osmotic pressure and hydrostatic pressure. All right. So we also have these things called impermeant particles, impermeant molecules. And basically they are substances that can't cross a cell membrane. We also call them non-penetrating solute particles. Basically they're trapped on one side of the cell membrane. And so think of this, the cell is this big and the molecule is this big. That's what we're talking about. Because of the size, or because of their polarity, or some ions will never be able to cross that cell membrane. So large proteins such as albumin. And again, all these things contribute to osmotic pressure. So as we have more things that are dissolved in the blood, we're going to have a greater osmotic pressure. As we have more impermeant particles, we're going to have higher osmotic pressure. So the greater the number of impermeant or non-penetrating solute particles, the greater the osmotic pressure. Hopefully that makes sense. If not, please let me know. All right, now osmosis. We already know that osmosis is the movement of water through a selectively permeable membrane. So up here, we talked about osmosis is the movement of water from a region of higher water concentration to a region of lower water concentration. But what we can do is flip that definition to say It's a movement of water through a selectively permeable membrane, so far nothing new, to a solution containing a higher concentration of impermeant solute. Does that make sense? So what I'm going to do is, well, we'll look at this in a couple slides. We have our osmotic pressure, is our pressure from everything that is dissolved or not dissolved in our bloodstream. We have our water, which generates hydrostatic pressure. And then eventually, hopefully, we'll reach this thing called osmotic equilibrium, where our osmotic pressure equals our hydrostatic pressure. All right, so we're going to skip this slide for a second. All right, so we have our selectively permeable membrane, and we have non-penetrating protein molecules that cannot cross the cell membrane. So, all right, so these purple protein molecules can't cross the cell membrane for this selectively permeable membrane for whatever reason. They're too big. We'll just say that they're too big. Now, what's going to happen is the water is going to have to move in order to dilute the proteins. So over here, if you count up the purple balls, we have 8 purple balls and 4 purple balls. Here we have 8 purple balls and 4 purple balls. So our ratio between 8 and 4 is still going to be the same. But if you we're not going to count up the number of water molecules. But if we did, the ratio of what we would find is that this would be at equilibrium between A and B over here. That the ratio of protein to water would be the same on A as it would be for B. So water is going to have to do the moving. And what we would find is that you have the protein pressure, or that osmotic pressure would pull the water up, and that hydrostatic pressure pulls the water, actually we'll do this side, hydrostatic pressure pulls the water down. So I hope that kind of makes sense. So go back to the definition. That first definition is movement of water from a region of higher water concentration to a region of lower water concentration. Yes, we did that. We moved from a higher water concentration to a region of lower water concentration, or we moved it to a solution containing a higher concentration of impermeant solute. We moved it from an area to a higher impermeant solute concentration. That's all we did. So we kind of flip-flopped that definition. All right, so in practicality, what we did is this is what happens in our cardiovascular system. And This is going to be an introduction. We're not going to worry about these numbers. We'll don't memorize the numbers, but basically, you know how we have arteries and veins and capillaries, and so we have arteries, and they actually shrink to arterioles and then into capillaries. Arterioles are basically just small arteries, and venules are basically just small veins. So this would be the arterial side of a capillary, and this would be the venule side of a capillary. So on an arterial side of the capillary, what we find is that we have a greater hydrostatic pressure than we do on osmotic pressure. So we're going to have an overall higher osmotic pressure. So what we're going to see is that we're going to find out that we're going to push water out of our blood vessels. And it's actually going to be about 20 liters of fluid per day. And then at the venule end over here, we're going to see that we don't have that much hydrostatic pressure, but we're going to have more protein pressure or osmotic pressure. So we're actually going to pull fluid in. We actually pull in about 17 liters of fluid per day. So 20 minus 17 is 3 liters. So actually we're left with 3 liters of fluid per day. And if we didn't have, let's say we had kidney disease or something like that, or protein deficiency, we might get edema. Edema is just fluid buildup. You see sometimes grandma's legs or ankles, and you can push, and she has pitting edema. But under normal circumstances, the lymphatic system would pick up this extra 3 liters of fluid, and it would get returned to the cardiovascular system. All right, so you'll see this slide next semester. We'll go over that really in depth. But I wanted to give you an introduction to this osmotic pressure and how fluid moves, because one, we'll talk about the movement of water here in the next slide when we get to tonicity. And two, I wanted to give you an introduction for next semester. So tonicity is basically how we compare what's happening on the inside of the cell versus what's happening in the blood plasma. If we have an isotonic solution, that just means the osmotic pressure inside the cell has the same osmotic pressure of our plasma. So we don't have a net gain or loss of water. And really you'll see this, if you have, you'll see hypertonic and hypertonic, with protein deficiency, or if you give a bunch of IV fluid rapidly. Now, a lot of times as a nurse, you'll probably just give, 0.9% normal saline or lactated ringers. That's going to be an isotonic solution. So really, you're going to make your patients pee if you kind of overdose them on that. Not much is going to happen. But there are times where you're going to give your patients hypertonic or hypotonic solution. Or if your patients do have kidney disease, or if you have an anemic patient where they don't eat a lot of protein, they might, if we have a hypertonic solution, what we see is that we have a higher osmotic pressure. So that means that there is a higher concentration of impermeate solutes compared with the cells that are in that solution. So inside the bloodstream, you're going to have a lot of impermeate solutes compared to those cells. So remember, the movement of water would go from the cells to try and dilute the proteins, that protein and all of those impermeate solutes, that other 8%, this other 8%, that we're talking about: proteins, electrolyte, waste products, nutrients, vitamins, hormones, gases. So hypertonic, more extras in the bloodstream compared to the cells. So water's going to move from the cell to try and dilute the extras that are in that bloodstream. So cells are going to lose water because they're trying to dilute whatever's in that bloodstream, and the cells are actually going to shrink up. In a hypotonic solution, we're going to have a lower osmotic pressure. We don't have a lot of, we have a lower concentration of impermeate solutes in that blood plasma compared to the cells. So we have more osmotic pressure inside the cells. So water's going to move from the plasma into the cells to try and dilute that osmotic pressure. So water moves inside the cells. Cells are going to expand, and possibly burst. We say that the cells lyse, L-Y-S-E. So very important. So we do have fluids out there that are hypotonic fluids, IV fluids, and hypertonic fluids. Hypertonic fluid, D10, D20, D50, dextrose 5%, or dextrose 10% in water, dextrose 20%, dextrose 50% in water. Hypotonic fluids, 0.45% normal saline. Dextrose 2.5% in water. So sometimes you'll be giving hypotonic and hypertonic IV fluids. So you have to be very careful about monitoring your patients when you're giving them these types of fluids. All right. So isotonic solutions. You have the exact same osmotic pressure, so nothing's going to happen. Your cells remain the same. In a hypertonic solution, remember, you're going to have more osmotic pressure inside the cell compared to the blood plasma. So water's going to rush into the cell to try and dilute that osmotic pressure. I said that backwards. I'm sorry. Hypertonic solution, you have more of osmotic pressure outside the cell than inside. So water's going to go from inside the cell to try and dilute what's on the outside of the cell, inside the blood plasma. So water's going to leave the cell, shrink. Hypotonic solution. You have a higher concentration of osmotic pressure inside the cell compared to the blood plasma. Water's going to rush into the cell to try and dilute the osmotic pressure inside the cell. The cell is going to expand and lyse or burst. All right, so cells are placed in a solution that causes them to absorb liquids until they burst. When water is removed from solution, newly cells added do not burst. The original solution was blank, while the second solution was blank. All right, so cells are placed in a solution that causes them to absorb liquids until they burst. So until they burst, that's a hypotonic solution. The original solution was hypotonic, and then the second part, newly added cells do not burst. So that's going to be an isotonic solution. All right, filtration. Again, passive process. No ATP is required. Basically, This is like using coffee grounds. For those of you who actually make your own coffee, water is just filtered or whatever is just filtered through gravity. It's used to separate solids from water or small particles from larger ones. Example, blood plasma leaves capillaries, water, small solutes are filtered, but large proteins are not filtered out. It just works by hydrostatic pressure only. There is no osmotic pressure associated with this. Only hydrostatic pressure. Think of water going through the coffee grounds. Hydrostatic pressure only until the pressure exerted at equilibrium due to the force of gravity. All right. Coffee grounds are mixed with water before being placed in a funnel filter. So we're thinking of filtration. The large grounds remained in the filter, but small particles passed through the filter. How did the small particles pass through? They move from an area of low concentration to an area of high concentration. Absolutely not. That would be an active process which requires ATP. They moved by hydrostatic pressure that was greater on one side of the membrane than the other. Okay, so far that's a good answer. Because filtration, hydrostatic pressure only. We're talking about a passive process, which is greater on one side than the other. That's down the concentration gradient. This one, answer C, talks about osmotic pressure. No, filtration is only hydrostatic pressure. They moved from an area of low hydrostatic to an area of high. Nope, that's an active process. So answer B is going to be our answer. All right, active mechanisms. Active mechanisms require ATP, cellular energy, in order to cross the cell membrane. We call that active transport, endocytosis, exocytosis, transcytosis. Basically, we are moving something against the concentration gradient. We are going from an area of low concentration or low solute concentration to an area of high solute concentration. It's very similar to facilitated diffusion, except remember, facilitated fusion requires those channels in order to move things in the cell membrane, except that we're going against the concentration gradient. We have to have those carrier molecules, those ion channels. Typically, we call them pumps. If you ever heard of the sodium-potassium pump, that's an example of what I'm talking about. All right, so we require ATP. That's going to alter the shape of that carrier protein, and that's going to drive those passenger particles through that cell membrane to the other side, where they are going to be released. All right, so sugars, amino acids, calcium, hydrogen ions, sodium potassium pump. Here's our equation. We will be talking about this equation a lot. ATP at adenosine triphosphate. So we have our adenosine molecule with 3 phosphates attached to it, going to ADP, adenosine diphosphate, plus our third phosphate plus energy. We're releasing energy. And then we have this statement here. We have this secondary active transport. Basically, it's a carrier transport that uses a sodium gradient to transport another substance across the cell membrane. But it says it's not an active process. It doesn't require ATP. We'll go into that here later on. All right, so here's an example of the sodium-potassium pump. Remember, we have a higher concentration of sodium on the outside versus the inside. We have a higher concentration of potassium on the inside compared to the outside. So what we're doing is we're going to use ATP to kind of charge or move this carrier molecule. In this case, it's called the sodium-potassium pump. And what we're doing is we're going to take 3 sodium molecules and 2 potassium molecules, and we're going to go against the concentration gradient. And we're going to move sodium outside of the cell to where there's more sodium, and we're going to take potassiums and move them inside the cell to where there are more potassiums. We're going to move them against the concentration gradient. We will talk about this more when we get to chapter 10. So sodium-potassium pump, active process, requires ATP. Now, I mentioned this statement, secondary active transport. So sodium coming in typically is only used for is facilitated diffusion. Facilitated diffusion is a passive process. Now, glucose, sugars, is going to be an active process. If we were only bringing in sugar, that would be an active process. If we were only bringing in sodium, that would be facilitated diffusion. But we have this thing called secondary active transport. Basically, what we're doing is we're going to bring in sodium and we're going to bring glucose in. Glucose is actually going against the concentration gradient. So technically that's an active transport because we're going against the concentration gradient. Sodium, remember, has a higher concentration outside the cell versus inside the cell. So here, sodium would be going normally facilitated diffusion, but we call this secondary active transport. Secondary active transport does not require ATP. So the body is good about not using energy when it doesn't have to. In this example, the body's already bringing in sodium under facilitated diffusion. So why not go ahead and bring in glucose as a hitchhiker? So sodium's going to be the primary facilitated diffusion. So go ahead and bring in glucose as well. If sodium doesn't require an active process, facilitated diffusion does not require active process, doesn't require ATP, then why not go ahead and bring in glucose as well? That's the only reason why we call it secondary active transport. Technically, it's an active process because we're going against the concentration gradient because glucose has a higher concentration. We're going on the inside of the cell versus outside, and we're bringing glucose in the cell, but We're not using ATP because glucose is just the hitchhiker in this scenario. We're already bringing in sodium, so let's just bring in glucose as well. So don't get confused when you see secondary active transport. Secondary active transport does not require energy, only because that primary molecule, in this case sodium, we're already bringing it in without ATP. That secondary molecule, glucose, is just a hitchhiker. All right, endocytosis. It's an active process. This is how we get things inside the cell. We have 3 different types of, basically these are for substances that are too large to enter the cell by diffusion or active transport. The cell kind of has to engulf these molecules. We have 3 types of endocytosis. We have pinocytosis, phagocytosis, and receptor-mediated endocytosis. Pinocytosis, cell drinking. Phagocytosis, cell eating. Receptor-mediated endocytosis is basically how cells eat with a receptor, or how cells bring in hormones, proteins, larger molecules. But we have to have receptors in order for those cells to engulf those hormones and proteins. So for phagocytosis, we have that's cell eating or bringing in solid particles. Remember I talked about bacteria. We have cells that are specifically phagocytes that eat that eat up stuff. But we also have monocytes that can act as phagocytes, macrophages, neutrophils, and dendritic cells that all eat up stuff. And they can not only they can remove bacteria and cellular debris. Okay, so this is pinocytosis and this is phagocytosis. And basically, we'll see that the cell starts to engulf the particle, and eventually the particles are inside the cell in a vesicle. Receptor-mediated endocytosis. Eventually, we have to, in order for something to happen, these proteins or these hormones have to link up with these receptors that are on the cell in order to bring that protein or that hormone inside the cell. A good example of this is how cholesterol gets inside the cell, or LDL, that low density lipoprotein, your good cholesterol. All right, exocytosis. Exocytosis is how we get rid of things from the cell. We saw that when we packaged up the protein vesicles and they fused to the cell membrane and dump out their contents. That's an example of exocytosis. We'll see this when we talk about releasing neurotransmitters from the cells. We'll see that in chapter 9 and 10. Or protein secretions from other cells. Well, we already saw that. When the ribosomes get packaged up in the Golgi apparatus and they form those vesicles, and those vesicles fuse to the cell membrane. That's exocytosis. Exocytosis is just taking this particle right here and going backwards. Exocytosis is just the opposite of endocytosis. This is exocytosis. We've already seen this example. That vesicle gets butted off. It goes to the cell membrane. It fuses with the cell membrane, and then that contents gets dumped out of that cell membrane, again, by exocytosis. It requires energy in order to do so. Transcytosis. Transcytosis, we really don't see this a lot. I just put it in here. I've done a lot of HIV work. So this is how HIV kind of starts. We see this, basically it's a combo. We see this receptor-mediated endocytosis followed by immediate exocytosis. So within the same cell. So the HIV will have receptor-mediated endocytosis, typically in the lining of the anus or the vagina, and then it will cross through that cell, followed by exocytosis. So it goes from one end of the cell to the other end of the cell into the person's bloodstream. So HIV attaches, it goes endocytosis, actually receptor-mediated endocytosis, and then it goes into the cell, and then exocytosis by ATP. It's an active process, and then it can attach to your white blood cells and replicate from there. All right, so this is just a summary slide of diffusion, facilitated diffusion, osmosis, filtration, the different types of active and active transport, endocytosis. We have the three types: pinocytosis, phagocytosis, receptor-mediated endocytosis, exocytosis, and transcytosis. All right, the cell cycle. You can see here that the cell cycle has a lot of different phases to it. We have this G1 cell growth, S phase, G2 phase. We're not really going to go into the cell cycle. We have this interphase followed by mitosis. We're really only going to talk a little bit about interphase, not much at all. That's, growth of the cell, maintenance of normal function. Mitosis, we'll talk about the division of the nucleus, and cytokinesis, which is actually the actual dividing of the cytoplasm, or the actual dividing of the cell. So like I said, we're not really going to talk too much about interphase. But I mentioned earlier that typically DNA is not visible under microscope. Really, there's no order to DNA. It's just kind of all crumbled up or all spread out in the nucleus. And then at some point in interphase, right before mitosis happens, this DNA kind of coils up into chromosomes, and it's basically complexed with this protein called chromatin. You can see that they are that they're wrapped around these things called histones, and obviously that's going to be stored in the nucleus. So and then also in interphase, that's where we replicate our DNA. All right, so humans have 46 chromosomes. And so we also have these things called chromatids. Chromatids are essentially, I'll just say, for lack of a better term, lines of the X's. So we have kind of one chromatid here and one chromatid here. So before DNA replication, we have 46 chromosomes and 46 chromatids because we only have one line of the X. And then after DNA replication, which occurs during interphase, we still have 46 chromosomes. And because we make two pairs of chromosomes, We actually have 23 pairs of chromosomes. But now we have 92 chromatids because we have one X over here, one side of the X over here, and we have one side of the X over here. So 43 times 2, or 46 times 2 is 92. And these chromatids, they're basically thread-like strands. They each contain that double helical structure. So there are two parts, each part of the X. Chromosomes are joined at this thing called a centromere. And then we have these things called telomeres. They're basically the ends of the chromosomes. They're considered the mitotic clock. And when we do mitosis, every time we replicate our DNA, a little bit of this telomere gets chopped off. And so we can only replicate our DNA for so long, which is why we die, or why cells die. Cells only have a lifespan of so long. Our taste buds only live like 3-5 days. Some cells live longer, but some cells can only reproduce so many times because every time we reproduce the DNA, a little bit of those telomeres gets chopped off. So that's why they're called the mitotic clock. All right, so mitosis. It says somatic cell division produces 2 daughter cells from an original cell. So we're not talking about reproducing our sex cells. So somatic cells, not our sex cells. We'll talk about that in chapter 22 next semester. So these are regular cells. And mitosis, division of the nucleus, via karyokinesis. Cytokinesis is the division of the cytoplasm. And we'll see that there's 4 stages of mitosis: prophase, metaphase, anaphase, and telophase. And I won't read them here because we'll cover them in each of the slides later on. All right, so prophase. What we see during prophase, that's our first phase. And in this example, we have 4 chromosomes, but remember that in humans we have 46 chromosomes. So here we start to see that cytoskeleton is disassembled. We don't see the microtubules. The DNA is already coiled up into chromosomes. We see that the chromosomes have condensed. We can see them under the microscopes. The chromosomes are genetically identical, and that they're joined at that centromere, where those 2 lines of the X cross. We see that the centrioles have moved to the opposite ends of the cell. Typically, they both reside at the nucleus, but now that they're at the opposite ends at the end of the cell, we can see that they've started to produce these spindle fibers. And eventually the spindle fibers will attach at the centromere. The nuclear envelope, or that nuclear membrane, starts to break down. Since nothing really has happened to the chromosomes yet, we still have 46 chromosomes, 23 pairs of chromosomes, and 92 chromatids. Prophase, metaphase. Metaphase is the next thing. Pretty much everything is gone. We don't see any organelles. We only see our centromere, or our centrosomes, our centrioles that produce the spindle fibers. They're attached to the centromeres of each chromosome. The spindle fibers are attached to the centromeres. The chromosomes are lined up kind of in the center of the cell. That's called the equatorial plane, or the metaphase plane, or the metaphyseal plane. Again, because the chromosomes have not done anything, they haven't separated, we still have 46, 23, and 92. Now, just remember that during cell division, chromosomes will not separate properly if the centrioles fail to attach to those centromeres. These spindle fibers have to attach to the centromeres in order for the chromosomes to separate. So prophase, metaphase, anaphase will be our third phase. This is where the chromosomes have separated. And they're going to start to be pulled to the opposite ends of the cell. Once they have separated, they are now individual chromosomes. They're genetically identical chromosomes because remember, we've replicated that DNA and now we've pulled them apart. The spindle fibers are shortening to pull those chromosomes apart. So now we actually have 92 chromosomes and 92 chromatids. We've pulled those chromosomes apart. So now we have 92 chromosomes. So prophase, metaphase, anaphase, telophase is our last phase. Basically, the chromosomes start to unravel. We really can't see them under the microscope anymore. It's going to return to that normal chromatin structure. The nuclear envelope starts to reappear. The nuclei starts to become visible. The organelles start to reform. Again, because we have not separated this cell yet, we have 92 chromosomes and 92 chromatids. Our cell has not officially separated yet. That's cytokinesis. We'll talk about that here in a second. All right, so here's just a review. Prophase, metaphase, anaphase, telophase. So I didn't put interphase in here because it's not really an event in mitosis. But if I were to, remember interphase is when we replicate the DNA. So if I were to include this, how I remember it is, so IPMAT, IP on the mat. If you have a mat in front of you, IPMAT, IP on the mat. Anyway. All right, so cytoplasmic division. We call that cytokinesis. It actually begins during anaphase and continues through telophase. What we see is that starting in anaphase, that the cell starts to kind of cleaves in on its side, and it starts to divide, and it continues through telophase, and then eventually that cell will split off into two. And once that cell splits off into two, then we will have 46 chromosomes and 46 chromatids. So if we go back up here into anaphase, technically anaphase should kind of look like this, where that cell starts to kind of cave in on itself. And in telophase, you can see how in telophase it's starting to cave in on itself. Then we have cytoplasmic division, where the cell really splits off into two. We have two new daughter cells that are hopefully genetically identical. All right. Then again, this is just a summary slide of how many chromosomes and chromatids that we see in each phase of mitosis, cytokinesis, and interphase. Here's a picture version. I just find that one a little bit more complicated. All right, stem cells and progenitor cells. If we actually looked at stem cells, so cells can only live so long. We talked about that. that mitotic clock and the telomeres. So, and they can divide so many times where they can't divide any longer. It's usually about 50 or 60 times. This is where stem cells come into play. Stem cells are cells that retain the ability to divide repeatedly without specializing. Think of stem cells as like a blank canvas. There are body's raw materials. This slide really doesn't do stem cells justice. An adult human has more than 290 specialized type of cells, including 14 types of cells that are unique to the embryo or fetus. The process of differentiation guides cell specialization by activating and suppressing the functions of many genes. So cell differentiation is a transition of a cell from one cell type to another. So no, you're not going to have to memorize this slide. So the three types of stem cells that we're going to talk about, the first one is going to be totipotent cells. So differentiation is a process of specialization of cells. We have these stem cells called totipotent cells. And basically, totipotent cells are self-renewal cells. They can divide to form two new stem cells. They can divide to form stem cells and progenitor cells, which we'll talk about later on. And they can give rise, or these totipotent cells basically are blank slates. They can basically go into anything. They have the capacity for self-renewal. They are, like I said, blank slates. They can become anything that they want. And if you remember from Psych 1010 or whatever, I'm sure you talked about the ectoderm, mesoderm, endoderm. They can divide into pluripotent cells or progenitor cells. Pluripotent cells are, like I said, ectoderm, mesoderm, endoderm. Progenitor cells, they can be lung cells or pancreas or cardiac muscle cells or neurons. Progenitors or pluripotent cells. So totipotent cells are kind of like the top of the hierarchy of stem cells. Like I said, blank slate stem cell. Pluripotent cells. They're the next high up. They can become a limited type of stem cells, belonging to either the ectoderm, mesoderm, or endoderm. Progenitor cells. So they can be the pluripotent cells. They can become, let's say, pluripotent. We know that you're going to be either the digestive system, urinary system, or respiratory system. Or we know that you're going to be the cardiac system, the skeletal muscle system, or the integumentary system. So we know kind of what one of three systems you're going to become. So again, not a blank slate, but still very broad in what that type of cell is going to be. Now, a progenitor cell, it's kind of third. It's still really high up, but it's third. It's lower than the totipotent and the pluripotent. So progenitor cells, they're still they're partially differentiated. So they can become any number of restricted cells. They're actually called a committed cell, in that they're committed to some organ system. So, for example, a neural stem cell can give rise to a progenitor cell that would end up becoming a neuron or a specialized neuron or a neuroglia that makes up neural tissue. So part of the nervous system. So now we're getting down to more of the system level. So here you can see, our stem cell, our totipotent stem cell, our progenitor cell, which will eventually be a neuron, or an epithelial cell, a skin cell, or a sebaceous gland, cell. All right, so I mentioned ectoderm, endoderm, mesoderm. Usually you talk about this in entry-level psych class. This is not a genetic embryology class. We're not going into this, but ectoderm will eventually become the skin, nervous system, or sensory. The mesoderm, you can see where the mesoderm goes: muscle, skeletal muscle, kidneys, circular, cardiovascular, lymphatic systems, reproductive system, or part of the skin. Endoderm, epithelial lining of the respiratory ducts, digestive, liver, pancreas, thymus, thyroid, parathyroid. So those are going to be your pluripotent stem cells that are going to go to your ectoderm, mesoderm, or endoderm. All right, so we do use stem cells to study and treat disease, typically for regenerative medicine. Regenerative medicine basically is how we use the body's ability to generate new cells to treat diseases and injuries. Bone marrow transplants, or how do we use reprogrammed differentiated stem cells. Remember that all cells have the exact same DNA. What we can do is we can actually kind of wipe out a bone cell back to its original stem cell, that totipotent cell, and regrow that cell to what we want it to be. And it's because of genetic expression. So each stem cell expresses a different subset of genes to make a kidney cell a kidney cell, a nerve cell a nerve cell, a liver cell a liver cell. And that's what regenerative medicine does. All right, cell death. So obviously cells have to die. We have two types of cell death. Apoptosis is basically programmed cell death. It's a normal part of development. It follows a very stepwise process, which we'll talk about here in a couple slides. Basically, it removes the webbing out of your fingers. When you're in the womb, your fingers are actually webbed. And it's going to remove the webbing between your fingers and your toes. It's going to sculpt your organs. And it's going to take away some extra brain cells. But it also peels away some of your damaged skin after you get a mild sunburn. Necrosis is a cell death from damage. And that's because your DNA, your cellular DNA, cannot be repaired. This is not a normal process. So think about grandma that is in the nursing home, that is in a bad nursing home, that has been left unattended to, and she's been laying on one side. And this is the reason why you have to turn your patients every 2 hours. If she's left on one side and she's on a bony prominence, like your hip, eventually that hip is going to lose blood supply and and that skin tissue is going to die. And you get a pressure ulcer or a decubitus ulcer. And that tissue is going to die and die and die. And eventually you're going to be left with a wound that is going to become necrotic, or necrosis is the noun form. Necrotic is the adjective form. And it might require surgical treatment. It's called debridement. So turn your patients every 2 hours. All right. So here's a picture version of apoptosis. So what happens during apoptosis? So we have these enzymes called capsases that are activated inside the cell. And these capsases basically they're just destroying enzymes that replicate and repair DNA. Basically, the cells can no longer replicate DNA. They no longer can repair DNA. They're going to activate enzymes that basically fragment the DNA and cut the DNA up into smaller pieces. They're going to dismantle your cytoskeleton, dismantle your nucleus, collapse everything. They're going to dismantle your organelles, destroy them, fracture your mitochondria, basically cut off your cell's energy supply. You're going to, these capsases, destroy or have the ability for the cell not to communicate with other cells. The cell needs to be destroyed. And they're going to release these enzymes that attract phagocytes. Remember, phagocytes eat up bad gunk, cellular debris. So they're going to attract phagocytes to come and break up that cell. And the body's going to reuse what it can and get rid of what it can't. All right. So that is the end of this chapter. If you need anything, please let me know. Otherwise, I will see you next chapter. All right, bye.